Hi, everyone. Welcome to Certara's Model-Informed Drug Development webinar series. Today is a special edition for Asia-Pacific regions on the topic, what you should know about FDA Project Optimus for your oncology drug approval. I'm Dr. Gailin Li, Senior Vice President of Certara, host this event in our Shanghai office. FDA Project Optimus is something everybody working on the oncology field should understand and what is about, what is the impact on your drug development strategy and the planning. Today, we will walk through and with our two speakers. Before we start the webinar, and I want to run quickly through some logistics. The webinar would take 60 minutes, including the Q&A. During the presentation, all attendees are muted, but you feel free to ask any questions, use the question panel during the presentations, or at the end of the presentation, you also have opportunity to submit your question via a quick surveys. After the presentation by two speakers, and our colleagues Amanda and myself would join you to answer your questions. Today, and Katrina, we are working through the general overview and the Project Optimus, and Amanda would provide the special considerations and the two case studies to share with you so you can appreciate how that has been implemented in your daily oncology development programs. So the first speaker, Kartika, and she has been working in the FDA for 3 years before she joined Certara. She also have some years working in Novartis and with focus on oncology drug development. And let's welcome Katrina for the first presentation. Katrina, please.
Good morning, everyone, and thank you for taking the time to attend. Dr. Mano and I are happy to be here today and to be speaking with you about the FDA's current expectations for oncology drug development and for dose optimization of oncology assets under the newly launched Project Optimus. We've organized this webinar today such that I will walk you through a general overview of Project Optimus, what the rationale behind its origin is, what the FDA's recommendations and expectations under Project Optimus are, and more importantly, what is required of drug developers in terms of planning for changes to early oncology development programs. Dr. Mano will then take you through some special considerations of high interest, such as combination development, rare indications and such situations, as well as some interesting case studies demonstrating the application of Project Optimus. By way of background, Project Optimus was launched by the Oncology Center of Excellence, or the OCE at the US FDA. Since its inception, OCE has launched several initiatives or projects intended to advance or improve the development process and the regulatory review of oncology products. These include projects such as Project Orbis, which many of you are already likely familiar with. Project Optimus is an important new OCE initiative that is aimed at reforming dose optimization in oncology in order to emphasize selection of dosing regimens of oncology therapeutics that maximize not only their efficacy, but also their safety and tolerability. Given its objective, leadership for Project Optimus at the FDA is provided jointly by the clinical and clinical pharmacology review functions at the agency. So that's something important that's worthwhile to bear in mind as you plan for Project Optimus related discussions and negotiations with the FDA. This slide summarizes several very informative references that document the FDA's thinking around Project Optimus, but perhaps the most useful place to start would be the newly released guidance, which is entitled Optimizing the Dosage of Human Prescription Drugs and Biologic Products for the Treatment of Oncologic Diseases, as well as this publication by Poziotinib et al., which provides more of the landscape behind some of the recommendations made in the guidance. The rationale behind Project Optimus comes from the difference between typical dose-response curves for traditional oncology chemotherapeutic agents and targeted therapies, which are shown on this slide. Traditional oncology chemo drugs had dose-response curves for efficacy and safety that essentially mimicked each other and in some cases overlapped, such that it made sense to use the highest dose that a patient could tolerate, in other words, the MTD, into further development as a recommended dose. The dose-response curves for targeted therapeutics, however, diverge, such that once target saturation is increased, further increases in doses do not necessarily lead to an improvement from a pharmacology or anti-tumor activity perspective, but could certainly lead to increases in safety concerns, for example, via the introduction of off-target toxicities. The current MTD-based dose-finding approach, which is still ubiquitously used in oncology, was therefore more suited to traditional chemotherapy agents, while the bulk of drugs in development today are targeted agents. Project Optimus is intended to rectify this mismatch and to shift oncology dose finding away from the MTD-based approach towards approaches that are more suitable to targeted agents, such that we can identify doses of these agents that strike good risk-benefit balance. A natural question that arises, of course, is why now? Why has the FDA chosen this particular moment in time to push for a more formal reform in oncology drug development? The answer to this question is no doubt multifactorial, but the most likely cause is a buildup of adequate experience at the agency to show that the post-marketing setting is not necessarily an acceptable or an optimal space to optimize dose. Post-marketing dose optimization studies can take a while to complete due to challenges with things like accrual, by which point a significant number of patients have already been exposed to a potentially inappropriate dose of the drug. Additionally, of course, dose optimization PMRs often lead to results that are equivocal or unclear, and that can be difficult to action from a regulatory perspective. Another key reason is likely increased patient advocacy and an increased focus at the FDA on patient voice. With an improvement in oncology drugs in development, patients taking targeted therapies are living longer and are therefore remaining on their drugs longer. These patients are now advocating to be able to stay on their drugs without significantly compromising their quality of life, and also for a better understanding of dose-response relationships so that they can make informed decisions with regard to their drug dosing. In other words, they would like to understand whether or not they absolutely need to tolerate a high dose of the drug for the promise of better efficacy, or whether it's okay to take a dose modification should one be needed. So what to expect under Project Optimus? The figure on the left side of this slide summarizes the typical dose-finding approach in oncology that's still very commonly used. This consists of a 3+3 dose escalation, usually in an all-comers solid tumor population, up to the maximum tolerated dose, or the MTD. The MTD, or sometimes the dose level below that, is then declared the recommended phase 2 dose, or the RP2D, and taken into expansion in various tumor-specific cohorts in order to identify a lead indication. And more often than not, it is that very same dose that ends up being taken into registrational studies. Under Project Optimus, this paradigm is expected to give way to lead to early trial designs that are intended to evaluate dose and efficacy response relationships with both safety and activity of the drug. Programs are expected to more thoroughly understand and account for later onset or cumulative toxicities past the DLT evaluation period, in particular toxicities that impact patient quality of life and those that impact or necessitate dose modifications. If there's one take-home slide I would like to recommend from my portion of the presentation, I would say it is this one. This slide integrates the bulk of the recommendations from the FDA relating to Project Optimus from a variety of different sources. The biggest change we should expect is the need for evaluation of safety and anti-tumor activity in an adequate number of patients at multiple dosage levels in the target population of interest, ideally in a randomized setting, ahead of any registrational study. The selection of these dosages should be based on sufficient preliminary understanding of the PK, PD, safety, and activity of these doses in the clinic, as well as an understanding of the translational appropriateness or relevance of these dosages. A good characterization of PK following multiple dosage regimens will be expected early in development. The agency has also indicated that there will be an increased focus on modeling and simulation activities, such as preliminary population PK and exposure response earlier in development. But at a minimum, this can be interpreted as a stage-appropriate integrated exploration of all available clinical data, for example, using simple techniques like graphical analysis. It will be important to consider toxicities beyond the DLT evaluation period and beyond DLTs themselves. Specifically, please consider the incidence rates of grade 3 toxicities, and in the case of some toxicities which have significant impact on patient quality of life, the incidence rate of grade 2 toxicities. It will be really important to understand the impact of toxicities on dose modifications and the timing of events, both of toxicity as well as activity relative to the start of treatment. In particular, there will be a high focus on symptomatic toxicities that can have a high impact on patient quality of life. To put this another way, this means that toxicities such as diarrhea and rash can no longer be written off or justified away as clinically manageable. In this spirit, the FDA is recommending collecting patient-reported outcome, or PRO, data starting as early as dose escalation studies. The agency acknowledges that the analysis of PRO information in early studies is largely quantitative and exploratory, but states that it can be a useful additional data source to complement traditional safety and efficacy information. There will be an expectation of all available clinical and translational data being integrated, including a preliminary understanding of dose and exposure response relationships in order to adequately justify doses that are brought into the next stage of development. So what does this practically mean, and how to implement the agency's recommendations in terms of a program design? This slide provides a general conceptual framework for the new approach to oncology dose finding. Drug developers are now being encouraged to adopt a more objective focused approach to early oncology study design, with the FDA signaling increased opportunities for more flexibility in these early stages. In general, however, early oncology programs would still include a dose escalation phase, if needed in all comers, or if possible in a more tumor-specific population. This would be followed by an initial backfill or an expansion at a few different dosage levels, ideally in a more narrowed tumor population, in order to collect a little bit more data to inform dose levels that should be tested further for efficacy and safety. This now would be followed by a more definitive dose optimization study, comparing 2 or more dosages in a meaningful number of patients to characterize exposure response and dose response relationships, which will allow us to select the dose that is brought forward into registrational studies. By this stage, by the stage of the dose finding study, the agency will expect to have at least a preliminary understanding of the food effect on PK for orally administered drugs, in order to inform selection of the dose with regard to food. Put another way, oncology dose finding is now expected to occur in 2 stages. Stage 1 involves collecting adequate information to enable selection of dosage regimens for evaluation in the definitive dose optimization study. Stage 2 is to determine the proposed recommended dose to be brought forward into registrational studies based on this definitive dose finding study. This is, of course, a general conceptualization, and there are certainly other approaches to trial design where some of these steps can occur more seamlessly, including, for example, leveraging information from the dose optimization study as part of the registrational package. So what is expected in terms of the design of this definitive dose finding study? Ideally, the dose optimization study should be randomized when feasible, and should include a comparison of at least 2 dosage levels. Cohorts in the study do not need to be statistically powered, but should be sufficiently sized to be informative based on an early understanding of anti-tumor activity. The doses selected for these studies should first and foremost be pharmacokinetically distinguishable. In other words, there shouldn't be a sizable overlap of exposures at the selected dosages. The high dose can be chosen with safety in mind. So this is sort of a no regrets dose that maximizes the chances of seeing responses and allows us to address the question of whether the high dose does indeed increase activity with acceptable safety and tolerability. The low dose can be selected based on available translational and clinical PK/PD data and should be the lowest dose expected to provide activity. In other words, the lowest fully active dose. Key information from this trial can be integrated with data from prior studies in order to characterize dose and exposure response relationships with efficacy. And this trial in particular should also include longer-term characterization of tolerability at the dose levels being evaluated. All being equal, that is, if the safety and efficacy are similar between the lower and the higher dosage, then the lower dosage should be selected as the final dose to be brought forward into registrational studies. Once data from the dose optimization studies is available, the selection and justification of the registrational dose should take a multifactorial approach. It is important to note that the FDA is looking for integrated evidence from the 4 key areas listed on the slide to support the proposed registrational dose. They are looking, in other words, at whether the totality of evidence up to that point, based on translational, clinical PK/PD, safety, and efficacy data, along with the dose and exposure response relationships, support the proposed recommended dose. Preclinical data can be used to define target saturation and narrow the range of dosages for further evaluation. In addition to objective response and tumor shrinkage type data, well-defined biomarkers can support an understanding of dose and response relationships with anti-tumor activity. The use of modeling and simulation early is encouraged, but the focus should really be on integration and useful visualization of clinical safety and efficacy data. This change in paradigm is expected to require a change in planning at the sponsor's end. An increased number of patients early in development will increase the amount of time potentially spent in this stage, which could lead to possible delays to a registrational efficacy study. There is expected to be an increased reliance on model-informed drug development, so please plan for appropriate resources early in development to enable such activities. There is also a potential need for increased regulatory interactions in order to obtain agreement on the doses we're bringing forward into the next stage of development. So when is it appropriate and optimal in order to engage with the agency on issues related to Project Optimus and dose optimization? The FDA has confirmed its willingness to engage with sponsors early in development and to partner flexibly with them in order to address dose optimization issues as development proceeds. Discussions regarding dosing with the agency can be initiated as early as the pre-IND meeting and can continue on through milestone meetings, other PDUFA meetings, at protocol submissions and amendment submissions, as well as via MIDD meeting. At the pre-IND stage, the focus would be on early dose escalation and expansion study design in order to ensure that the early development plan will provide sufficient information to determine dose levels that will be evaluated further for efficacy. At the end of phase 1 meeting, the focus will be to obtain agreement on the dosages selected for further evaluation in the dose optimization study. At this point, you would need data from a minimum of 6 to 10 patients at a few different dosage levels, an understanding of PK and where possible PD properties across dosages, including a preliminary understanding of food effect on PK for orally administered drugs would be important. An understanding of where clinical exposures lie relative to targets derived from translational pharmacology study, as well as a preliminary understanding of safety and tolerability across dosages, and enough information on anti-tumor activity to define an appropriate low dose will be expected. At the end of phase 2 meeting, the goal would be to obtain agreement on the doses selected for the registrational study, by which point you will need a full justification of the dose, including a good body of safety and activity data at a minimum of 2 dosage levels. In order to help maintain consistency of arguments across all these FDA interactions and to aid with integration of data, we strongly recommend that sponsors consider building a dose justification white paper that can be updated and submitted to the agency as the drug moves through development. I would also like to take this opportunity to point out that the FDA has indicated that lack of data or inadequate dose justification at critical milestones could lead to an increased possibility of clinical hold on further studies, as the FDA may determine that patients in future studies are exposed to unreasonable or significant risk, or that there is inadequate data to determine risk, or that the design of the trial is deficient to meet its stated objectives. With that, I'd like to pass this over and transition to Dr. Mano, who will take us through the second half of this presentation. Thank you for your time and your attention so far.
Thank you, Dr. Katrika, and thank you for everyone submitting your questions. Let me now invite our second speaker of today, and who is Dr. Amandine Menon, and she's our colleague in the Certara. And Amandine has been working in the industry as a clinical pharmacologist for over 15 years, with a wealthy experience on oncology drug development. Hi, Mandy. You can start your presentation.
Good afternoon, good evening to all attendees. Thank you, Dr. Lee, for the kind introduction. Thank you, Dr. Shetty, for nicely introducing Project Optimus and how it will impact the dose optimization of oncology drugs. Now I will share with you some special considerations and also 2 case examples. Here I will go over the main questions we get from clients. And the sample size for the dose finding cohorts or trial is probably the #1 question. And it's confirmed by the questions we received. A lot of them were about sample size. To be honest, no one size fits all, but FDA has been recommending around 20 to 40 patients per dose level, but it can slightly be decreased if you evaluate more dose levels. Now moving to dose optimization for combination. Combination dose optimization is impacted by multiple factors, such as the mechanism of the combination. Is it additive or synergistic? It's also impacted by the drugs in combination. Are they new drugs, or is one or the 2 drugs already approved? And if one of the drugs is already approved, is it approved in the indication of interest or in another indication? Also, has its dose been dose optimized? So all these factors will impact the complexity of the combination dose optimization. Another important topic is what about rare indications? So even outside of oncology field, selecting the dose or the endpoints is a difficult topic for rare diseases, but still standard for regulatory approval remain the same. So for oncology, the recent guidance on dose optimization does not include any specific advice or free pass for orphan diseases. It means that Project Optimus principles are applicable to rare indication as well. So for this drug, there will be a challenge to wisely include patients in the studies and leverage, for example, adaptive designs or Bayesian-guided dose escalation to, like, let's say, save patients. Also, a question that comes back often is how to extrapolate or bridge the dose from an indication to another. So here we can leverage knowledge from tumor agnostic programs. You have to consider both non-clinical and clinical data. So, for example, is the mechanism of action independent of the tumor type? Is the target expression similar? How is the PK? And what about the safety? And is there any overlapping toxicity with one of the combination drugs, which may be different from another indication? So all of this will help you to figure out if the dose that was optimized for your initial indication is applicable to another indication. Also, a concern for a lot of sponsors is what about dose selection for drug with limited monotherapy activity? In that case, the question is it mandatory to optimize the dose in monotherapy before moving to combination, knowing that we don't expect any monotherapy activity? Honestly, this is still an evolving area and there is no single recommendation. It's more on a case-by-case basis. If you have other drugs of the same class that did not show any monotherapy activity, it will make it easier and you may be able to move to combination optimization early in the development. If you don't have any class evidence, you may then rely on non-clinical data, but you may need to generate some monotherapy data anyway. So whatever the plan, as explained earlier by Dr. Shetty, the plan needs to be discussed with the FDA as early as possible. And finally, how to leverage class knowledge. So it's, for example, the well-known PD-1, PD-L1 inhibitors, but it's applicable to all classes of drug. You can use class knowledge like binding affinity comparison or trough target concentration from competitors to support and ease your dose selection, but it won't prevent you to dose optimize your own drug. Now, turning to the first case study. It's the example of Asciminib, a tyrosine kinase inhibitor approved by the FDA in October 2021 in chronic myelogenous leukemia. So even if the drug was approved before Project Optimus was officially announced, it shows the application of sound dose optimization in a drug development program. The first lesson from this program is what knowledge can be leveraged from in vitro, in vivo pharmacological data. Here, for Asciminib, there was a clear difference in the IC50 in wild-type BCR-ABL cells and mutated BCR-ABL cells. And the same finding was observed in xenograft mice tumor model. The wild-type tumor model treated at 30 mg/kg BID had a tumor regression of 92%, whereas the mutated tumor model treated at the same dose had a tumor regression of only 56%. It means that probably the wild-type population will require lower doses than the mutated population, and that the dose may need to be optimized separately for wild-type and mutated populations. The second learning from the in vivo model is that the ID regimen improves the tumor regression compared to QD regimen. If you look at the results, the tumor regression when the tumor model was treated with 30 mg/kg QD for 11 days was 33%, whereas the same model treated at 30 mg/kg BID for 7 days had a tumor regression of 92%. So a major difference in tumor regression. And the last learning from the in vivo data is that tumor shrinkage was similar and maximum from those starting from 7.5 milligram per kilogram BID, showing that in that case, more drug does not really do better, and there is no need to push too much the dose. So we can learn a lot from these experiments to inform the design of the clinical trials. And that's what the sponsor did. Based on the pharmacological finding, BID dosing was explored in initial cohorts, and they started the QD cohorts later on based on emerging PK and preclinical data. And also, the wild type and mutated CML patients were explored separately in dose escalation and expansion, and with different dose levels. Another interesting learning from Asciminib program. So here is the design of the phase 3 study. Asciminib 40 milligram BID was evaluated versus standard of care, but they were able to get 2 dosing regimen approved in the USPI, 40 milligram BID and 80 milligram QD. But 80 milligram QD wasn't tested in phase 3. How did they do that? They actually leverage modeling and simulation, and especially exposure response, to get a new dosing regimen approved without testing it in a confirmatory trial. They leverage first the exposure efficacy relationship, and were able to generate the predicted MMR rate at week 24 for both regimen. And you see here that the MMR rate is very similar between 40 milligram BID and 80 milligram QD, 26.3% versus 25.8%. Moreover, the exposure safety relationship showed that there was no association between safety events and increase in PK metrics. So for example, if you have an increased Cmax with 80 milligram QD, which is likely, you don't expect more safety events. So based on this robust exposure efficacy and exposure safety relationship, they were able to justify and get the approval of the 80 milligram QD regimen, even if it was not tested in the pivotal study. So Asciminib is a very good example of how you can leverage non-clinical data to inform clinical trials, but also how ClinPharm and pharmacometry can help to inform your dose selection process and thus speed up your development and also increase your probability of success. Now, the second example is the example of Poziotinib that shows how FDA currently applies Project Optimus principles to NDA review. So Poziotinib is also a tyrosine kinase inhibitor with a proposed indication in HER2 exon 20 plus non-small cell lung cancer. The sponsor filed for accelerated approval, and then an ODAC meeting took place in September 2022. The outcome of the ODAC meeting was negative, and then the FDA issued a complete response letter with denied approval in November 2022. But why? So the sponsor filed for accelerated approval based on cohort 2 of their NIT 20 trial. But the FDA raised issues. So the key issue was that efficacy was not improved with Poziotinib over current therapy. But the FDA also cited inadequate dosage optimization as a key reason for denying approval to the drug. Indeed, the recommended dose was based on a limited data in a very limited number of subjects, and the recommended phase 2 dose was selected as the dose level below the MTD being 18 milligram QD, the recommended dose being 16 milligram QD, without sufficient justification. Moreover, exposure-response relationship did not support the proposed dose. The efficacy data were inconclusive and did not show that increasing the dose would do better in terms of efficacy. And the exposure-safety relationship showed that treatment emergent adverse events were correlated with an increased average concentration. So for Poziotinib, if you push the dose, if you increase the dose, it will translate into a higher average concentration and thus a higher risk of treatment emergent adverse event. And that's what happened probably with the 16 milligram QD, as most of the patients had dose interruptions or dose reductions. Here you see 83% of the patients had drug interruptions and 57% of the patient had dose reduction. If you look at the plot here, beyond 24 weeks, most of the patients actually received less than 12 milligram QD instead of the initial dose of 16 milligram QD, showing that the proposed dose was probably not tolerated. In parallel of cohort 2, the sponsor tested alternative dosing regimen in exploratory cohorts. So they tested the BID dosing regimen from 6 to 10 milligram BID and other dose QD from 10 to 16 milligram QD. Overall response rate appears pretty similar between the different cohorts. If you look here at 12 milligram QD, 8 milligram BID or 16 milligram QD, it's similar. So again, it seems to show that more drug may not really do better with Poziotinib. So the sponsor took these findings into account, and now the ongoing confirmatory trial has started with a dose of 8 milligram BID instead of the initial proposed dose of 16 milligram QD. So based on that, we may be able to see later on if the split of the dose was able to mitigate the toxicities of the drug. Because, yeah, the FDA now scrutinized the recurring toxicities. In the case of Poziotinib, the FDA noticed higher rates of symptomatic toxicities such as rash, diarrhea, compared to other TKIs. So for example, if you look at the plots, the incidence of rash was more than 90%, and for the diarrhea, it was around 80%. So it shows how now the FDA reviews the safety profile of the drug during the NDA process, and there is clearly a shift towards less acceptance of low-grade toxicities that impact patients' quality of life. This focus on recurring toxicities that impact patients' quality of life is also correlated with the recommendation by the FDA in the recent dose optimization guidance to include patient-reported outcomes in all trials. So they indeed recommend to include PROs, so patient-reported outcome, in all trials, including early-stage trials, and the objective is to better incorporate patients' voice in drug development and dose selection. In the case of Poziotinib, they noticed that the PROs were not adequately assessed. So this case example really shows how the FDA now reviews the dose selection at NDA stage with a focus on recurring toxicities, patient-reported outcome, and how the dose was optimized. Now to conclude, here are the key take-home message from Project Optimus paradigm change. So we have to move away from traditional MTD-based oncology dose finding paradigm. We have to plan for additional resources in early oncology programs, so to support clinical trial conduct, but also for robust stage-appropriate data analysis, like dose response, exposure response, PK/PD analysis. We have to plan also for regular FDA interactions related to dose selection. We have to switch to objective-focused early oncology trial planning and nomenclature. And last but not least, dose selection is multifaceted and requires integrated analysis of all available data, so including obviously PD, anti-tumor activity, safety, including dose intensity, dose reduction, dose delays, also PK, and also that is pretty new in oncology, patient-reported outcome, even in early stage trials. And here I want to present you the Certara's Project Optimus team. So it's a team of 9 very experienced clinical pharmacologists and pharmacometricians. And it's led by Jolly Bullock, who is a former FDA Clean Pharm reviewer and executive VP head of the clinical pharmacology team here at Certara. So far, we have supported more than 80 assets to navigate the new Project Optimus requirements. So we have a good database and good understanding of the FDA requirements. And if you need for your own project to navigate Project Optimus requirements, we can help. So for drug at pre-IND stage, we have what we call the Optimus Fitness. For drugs at a later stage of development, like end of dose escalation or prior to start the pivotal trial, we can propose Project Optimus gap analysis. But we can also take part of workshop seminar, we can help with FDA interaction, and we can also help with questions raised at the time of NDA BLA submission if, unfortunately, there are some questions regarding the dose optimization raised at that stage. So thank you for joining our webinar today, and that's the end of my presentation.